Thank you very much for this great opportunity to be part of this exciting symposium. I'm Dr. Marakalala, and I'll be talking to you about proteomics of TB granulomas. And granulomas are basically structures in the lungs that are containing bacteria at the centers, and usually granulomas have macrophages which are surrounded by giant multinucleated epithelioid cells, which is surrounded by a lymphatic curve that has mostly T cells and B cells. And in this intact structure, granuloma can limit mycobacteria at the centers here. However, this containment can actually be tempered with when granulomas collapse, and a simple dissociation of granulomas has normally been characterized by a formation of necrotic material at the center known as caesium, which can actually lead to liquefaction, and liquefaction resulting in cavitation. And it's through cavities that bacteria can disseminate throughout the lung. And also, a cavity that is exposed to airways can lead to coughing of bacteria, leading transmission of TB. So it's really important to understand events that lead from a normal containment of a solid granulomas to damages associated with caseation and eventually cavitary TB. So basically what we are interested in here is answering a question on what exactly drives granuloma changes that are associated with TB damage or TB progression through lung damage. So to answer these questions, we basically looked at tissues of the lungs from patients who were treatment refractory and were treated by surgery. So we basically looked at solid granulomas and then caseous granulomas, as you can see a caesium here, and also cavitary granulomas. So we dissected specific regions in these particular tissues using laser capture microdissections, and we hydrolyzed the samples and then used mass spec, and then using a specific program called MaxQuant, we're able to map specific peptides to particular proteins to give us total proteomes of each granuloma types. And basically here is just to take you through images where we had a solid and cavitary granulomas dissecting the cellular regions and also cellular regions in this area. And in the same way, when you have a caseous granulomas, we actually dissected these caseous regions here and also dissected what remains after the caesium has fallen in the cavitary granuloma, as you can see here. So our proteomes actually showed that caesiums from both cavitary and caseous granulomas or necrotic regions were clustering together, whereas cellular regions, regardless of the source of granulomas, were actually clustering together. But what was really interesting when we show here the volcanic plot, we saw that inflammatory materials were highly associated with caesium's form of granulomas, whereas actually cellular materials were associated with proteins that are involved in biogenesis, such as protein synthesis. And some of the exciting molecules that we saw that were related to pro-inflammatory responses were molecules that are involved in processing of arachidonic acid. Arachidonic acid can actually be broken down, leading to leukotriene and lipoxins through lipoxygenases, or on the other arm, it can be utilized by COX-1 and COX-2, leading to production of prostanoids. So basically, some of the molecules we saw were actually in this pathway that lead to pro-inflammatory leukotrienes here. And as you can see here, ilox-5, that is upstream of leukotrienes, and LTA4H, that leads to inflammatory eicosanoids such as leukotriene B4, were highly enriched more in the caesium regions of both cavitary and caseous granulomas as opposed to cellular regions. So this was really exciting, but we needed to validate the trueness of this particular data. So we used immunohistochemistry staining by utilizing antibodies specific for LTA4H. And as you can see here, this is basically a granulomas where you actually have a caesium here. So in this small caesium surrounded by a solid material, we saw that by staining you can see high level of intensity of LTA4H in the centers of the caesium and the surrounding regions where macrophages seem to be undergoing breakdown by necrosis. And on the surrounding cellular regions, however, this protein was very little, showing that LTA4H, that is pro-inflammatory, is associated with caesiums. And you could also use another granuloma here, like a cavitary granuloma, to show that here the region that is solid has less representation of this LTA4H. However, necrotic material that remains after the calcium has fallen is more highly represented with LTA4H. So this was really exciting that we actually went on to characterize the total granuloma types to measure the amount of intensity through this expression, and we found that LTA4H was higher in the total caseous granulomas and cavitary granulomas compared to solid granulomas. So far we basically used LTA4H and its validation through immunohistochemistry to unpack the pathway of arachidonic acid. So as such, we're really interested on seeing what happens on the other side, the other arm of arachidonic acid. And we found that looking at COXes, COX1 and 2, starting with COX1, we found that actually COX1 compared to LTA4H was little at the center compared to the actual solid material, where LTA4H was very little. So basically this is opposite expression of what you see on the other arm of arachidonic acid. This was so interesting to us that we also wanted to measure the total expressions or representation of the COXes in different granuloma types. And here you can see in the cavitary and caseous compared to solid granulomas, the COXes were lower in cavitary and caseous compared to solid, still opposite of LTA4H. So this actually suggesting that products of COXes might be actually limiting the high inflammatory effect that is caused by the inflammatory eicosanoids such as leukotriene B4. So basically one of the known molecules that are anti-inflammatory in the COX pathway is this stable form of prostacyclin, which we think might be limiting the uncontrollable levels of leukotriene that leads to damage. And we used MALDI-MSI to measure or to quantify the amounts of this prostacyclin, and we found that it was associated with cellular layers, and it was actually less so in the calcium. So this suggesting that you can actually use MALDI-MSI to quantify products which are actually in particular pathways that are utilized by specific enzymes that we saw in proteomics and we validate with immunohistochemistry. So you have immunohistochemistry, you have proteomics, and you have MALDI-MSI that are agreeing to specific pathway. So with MALDI-MSI in mind, this is just an example. So how MALDI-MSI works, you can have a slide with the tissue, and then we measure a mass spectrum of particular metabolites. In just one slide, you can actually view different things. And here, for example, when you have a granuloma, a lung that has a solid form here in this image, go in there, it's a solid form of a lung, and this is where the calcium begins. So you can actually see different molecules distributed differently on the same region. For example, arachidonic acid here seems to be really more distributed on the surrounding regions of the calcium. So what this tells is that arachidonic acid is really higher on the calcium, and depending on what pathway in either the COXes or lipoxygenase utilize them, they can actually give you the fate based on the level of inflammation. But what's really interesting also is that these molecules such as LTA4H can then be, since they lead to damage and they've been really associated in diseases such as TB meningitis, can also be utilized in other samples that have TB samples such as spine TB and so on. And such molecules could also be targeted for host-directed therapy with proper understanding. I really thank you for this opportunity to present here, and I'll be happy for questions and discussions afterwards. Thank you.